A special type of protein is the enzyme, though not all enzymes are proteins. It is thought, through understanding the RNA world hypothesis, that proteins assumed the catalytic function of RNA. And now RNA is utilized through translation to synthesize proteins that fulfill that role. An enzyme is a substance produced by an organism and acts as a catalyst to bring about a specific biochemical reaction. Due to the specific sequence of amino acids, the interactions between them typically create a globular structure, having alpha helices and beta sheets. These foldings create various features and landmarks on the enzyme. One is referred to as the active site, where substrates or competitive inhibitors may bind. If a substrate or substrates bind to the active site, the enzyme speeds up the reaction rate by decreasing the energy required to initiate the reaction, or the activation energy. The product or products are formed. If bonds are created, causing energy to be stored, it is an endergonic reaction. In these reactions, there is less energy in the reactants than there are in the products. If bonds are broken, causing energy to be released, it is an exergonic reaction. In these reactions, there is more energy in the reactants than there are in the products. If a competitive inhibitor binds to the active site, it blocks the substrate and prevents the catalyst from performing the reaction. Increasing the substrate concentration could still result in the maximum rate to occur. Other regions that may bind to substances are allosteric sites, basically anything that is not the active site. These are where noncompetitive inhibitors bind. When they do, they cause a physical change in the active site. This blocks the substrate and prevents the catalyst from performing the reaction. Increasing substrate concentration will never result in the maximum rate to occur. One enzyme in particular, lactase, happens to be one that has a different frequency amongst various populations. According to the National Institutes of Health's National Library of Medicine, approximately 65% of the human population has a reduced ability to digest lactose after infancy. Lactose intolerance in adulthood is most prevalent in people of East Asian descent, affecting more than 90% of adults in some of these communities. Lactose intolerance is also very common in people of West African, Arab, Jewish, Greek, and Italian descent. It was quote-unquote normal for humans to lose lactose tolerance or expression of the lactase gene. When we no longer needed our mother's milk to survive, the gene became deactivated. However, around 7,000 years ago, a mutation occurred that continued the expression throughout adulthood in European populations. Migration, trade, and the domestication of the cow in the Middle East and North Africa allowed dairy to be a source of nutrition. This mutation proved to be beneficial and spread throughout the descendants of Europeans, Middle Easterners, and North Africans. However, this mutation is not found in all members of these populations, and some populations have significantly low frequencies of the mutation. We as humans consume dairy as a source for calcium. However, those with lactose intolerance have a much more difficult time digesting lactose. Signs and symptoms of lactose intolerance include diarrhea, nausea, and sometimes vomiting, abdominal cramps, bloating, and gas. There are medications or pills you can take before consuming dairy products to help you with digesting this disaccharide. However, many food sources have significant amounts of calcium to support a dairy-free lifestyle. These include broccoli, edamame, figs, oranges, sardines, canned salmon, white beans, and almonds. This created a market for lactose-free products, including foods, medications, and treatments. In order to create lactose-free products, you must immobilize the enzyme. Immobilized enzymes are when these protein catalysts are permanently attached to a surface. This can involve alginate beads. Alginate beads are composed partly of sodium alginate, a water-insoluble, gelatinous, cream-colored substance found in the cell walls of brown algae. When sodium alginate reacts with calcium, it becomes insoluble and solid, or gelatinous. The enzyme lactase either becomes entrapped inside the beads or fixed to the surfaces during this reaction. Then, milk can be passed over these beads, creating lactose-free products. The advantage to using calcium alginate beads is that it is more resistant to changes in pH or temperature. Additionally, it allows you to control the specific reaction that will occur without contaminating your product. For example, it is possible to create a genetically modified yeast or bacteria that synthesizes lactase. Rather than introducing the microorganism into a milk supply, you can purify a sample of lactase and add that only. This ensures food safety. The disadvantage to alginate beads is, if enzymes are entrapped in the substance, the reaction rate will be lower than the maximum. The advent of lactose-free products offers several advantages. It allows people who are lactose intolerant to consume dairy. Second, lactase catabolizes lactose into galactose and glucose. These monosaccharides taste sweeter than lactose, reducing the need for additional sweeteners. These two monosaccharides also happen to be more water-soluble than lactose, giving smoother texture in ice cream. If you are trying to produce a dairy product that requires fermentation, like yogurt, then bacteria can ferment glucose and galactose more rapidly, shortening production time. Not only do immobilized enzymes have a role in beer and dairy production, but they have a wide array of uses. It is a technique used in paper production, biofuels, medicine, and biotechnology. The enzymes can be reused over and over, especially when pH and temperature are controlled as to not denature the enzymes and also achieve the maximum rate. And you can also produce a much more purified product by not allowing the enzymes to remain blended with the product. How do we know what the optimal temperature and pH for lactase are? Are these optimal ranges the same for all people? We know what we know about enzymes because of experimental design. Researchers, innovators, and inquirers needed accurate quantitative measurements in enzyme experiments. For example, lactase functions best around body temperature and in slightly acidic conditions. As you deviate away from these conditions, the activity of the enzyme decreases. This is because researchers designed experiments that tested a wide range of temperatures and pH to measure the activity or rate of reactions. In experimental design, it is imperative that you begin with a solid research question. This needs to be clear, focused, and relevant. It should also somehow indicate what variables you are going to test. For example, a bad research question about enzymes would be, how do enzymes work? The reason this is a bad question is because it is neither clear nor focused. What enzyme is being tested? What condition are you manipulating? How is data going to be collected? Are you directly or indirectly measuring the activity? Aside from all of this, it's not really a relevant question for the purpose of research, though it may be a question you genuinely have. Why is it important to understand how enzymes work? A better question would be, to what extent do alkaline environments affect lactase activity? This is clear and focused as to what you are going to test. The pH will be above 7, and you are measuring lactase's ability to catabolize lactose into glucose and galactose. You can then explain the relevance of this experiment through research. The research question then helps you write your hypothesis and identify the variables. In this experiment, I would set it up like this. I would test lactase activity at a pH of 6. This is my control group and allows me to confirm with my research that this is indeed the optimal condition. I would then test lactose in different pH environments. These are my experimental groups, and I would choose to test it from pHs between 7 and 14 at regular intervals. The way I would design my experiment is so that I can collect quantitative data for statistical analysis and determine how similar or different the experimental and control groups are to each other. In this example, I could use glucose test strips, but these are only going to indicate whether there is glucose present. To collect quantitative data, I would then take the solutions and measure glucose metabolism rates by yeast. However, living things and the substances they produce have variation. Not only would I test the activity of lactase at each pH, but I would repeat trials to ensure that I collect reliable and consistent results. A valid result should be able to be replicated independently, whereas an invalid result, originally achieved due to some error or perhaps just chance, will not be able to be consistently reproduced. Therefore, replicates account for the variation of living things, and processing the data through statistical analysis will reveal any patterns, trends, and discrepancies in the data. And this is why replicates are the most important. Statistical tests are strengthened by the degrees of freedom. Degrees of freedom are based on the sample size and the number of independent variables. We will only design experiments with one independent variable, so we will use degrees of freedom only for the sample size. Each statistical test calculates degrees of freedom differently. In this hypothetical example, my sample size is the total number of trials performed at each pH. Trust me, I understand this is totally abstract, and it takes practice with actual materials to truly develop meaning. We will be performing at least one enzyme investigation in class and include all of the above considerations in our experimental design.